What are the main ingredients for scientific research? According to the Nobel Prize winning Paul Ehrlich, these were Geduld, Geschick, Geld, and Gluck. Patience, ability, money, and good luck. And certainly this high-achieving man needed all of them. He was to be the director of the Royal Prussian Institute of Experimental Therapy in Frankfurt, and in 1908, the joint winner with Eli Metchnikoff of the 1908 Nobel Prize for Medicine for his major contribution to medical and biological research in the field of immunology. You would think that for this academic brilliance, he would have been a wonderful student. Quite the opposite. He hated exams, well, don't we all? And was reluctant to study any subject he did not find challenging. Luckily, chemistry did interest him, and he became passionate in his study of that subject. In his doctoral thesis at Leipzig University, he studied histological staining and its relationship to the dyeing of textiles. This was a subject that fascinated him so much that the dyes he used for staining marked his hands, clothing, and could be found all over his laboratory and his lodgings. I'm sure his landlord or landlady welcomed that. Now, in this work, he was helped by his cousin, the bacteriologist Carl Weigert, who developed his own methods of staining bacteria in tissue sections for study under the microscope. And he had contacts with the German chemical industry, which at that time was producing a large number of aniline dyes. Ehrlich observed that specific chemicals interacted with particular tissues, cells, or microbial agents in both stains and dyes. The result of this was that not all dyes stained everything. It suggested to him that chemical structures could not affect a cell without becoming attached to it in some way, and that there could be no reaction unless the dye attached itself to a receptor on the chemical structure. Having first established this idea of receptors, he then looked at the possibility that if there were dye receptors, there might also be similar chemical structures that might act as drug receptors. And this led him onto a search for chemical subjects, chemical substances that would act on microbes, but not on the human body tissues. He was looking for a magic bullet. Paul Ehrlich next turned his attention to the development of a fast method of staining the newly discovered tubercle bacillus, only 6 months after it had been discovered by Robert Koch to be the cause of tuberculosis. The result of his passion for this work led to Ehrlich discovering that he was suffering from TB, which meant that he had to give up work for 2 years and seek a cure in the drier climate of Egypt. Then, on his return to Berlin, he took up an unpaid position in Paul Koch's new Institute for Infectious Diseases. In 1892, he began what was to be a very unhappy collaboration with Emil von Behring and Shibazaburo Kitasato. They were working on the development of vaccines against diphtheria, then a major cause of childhood death. And also they were working on vaccines against tetanus. In this work, Ehrlich's very precise and painstaking methods of quantifying and standardising the antiserum were crucial. However, it all turned out badly. Behring reneged on a promise to include Ehrlich in a commercial contract for producing the resultant diphtheria antiserum. This contract was to bring a fortune to Behring and to lead to a very acrimonious split between the two men who had been born a day apart. They never saw each other again after this quarrel. Ironically, on the centenary of their joint births, they were both commemorated on the same German postage stamps. Ehrlich, though, now moved on, and in 1896 was appointed to the prestigious post of director of the newly established Institute for Sera Research and Sera Therapy, which moved to Frankfurt, having originally been located in Berlin. For the first time in his life, he had a proper salary and adequate research funding. The main purpose of the Institute was the state control of therapeutic sera, and Ehrlich was able to use his work with von Behring's anti-diphtheria serum to standardise sera in units related to a fixed and invariable standard. This was to be the basis of all future standardisations of vaccines. However, he also had the opportunity to carry out more original work in the field of immunology. He was determined to understand how the tetanus antitoxin worked. And in order to do so, he advanced a series of hypotheses that formed the basis of his revolutionary side chain or chemical affinity theory. He proposed that cells have specific receptors for antigens and that they shed these receptors in the blood when they come into contact with the antigen. He compared this process to a key fitting a lock, with each molecule of a toxin combining with a particular amount of antitoxin. The tetanus toxin, for example, would thus become bound to the cells of the central nervous system by attaching it to the chemical side chain of the cell nucleus, thus blocking their functions. The cell would then produce new side chains to replace those that had been blocked. These were the antibodies produced by the action of the toxin. Well, it was a revolutionary theory and was based on the idea that these reactions were chemical in nature and that they involved agents that were toxic to specific bacteria but would have no effect on the host. In effect, the antibodies would act as magic bullets which find their target by themselves and would be capable of killing only the specific organism that had invaded the body. Just as the antibodies in blood serum engulfed the bacteria that could target a particular infection without harming anything else in the human body. On one occasion, he put this these ideas before some of his friends on the night train from Berlin to Frankfurt. These were the British pathologists William Bullock and Almroth Wright. They made so much noise that the conductor thought they were arguing and threatened to put them off the train if they didn't quieten down. Now, for Ehrlich, the real challenge was to find tailor-made chemicals capable of acting against particular infective bacteria. His first experiments involved the use of methylene blue, one of the aniline dyes originally discovered by W.H. Perkin. And he found this was a quite promising use against malaria. So he turned his attention to finding a form of chemotherapy that would target the trypanosomes that caused sleeping sickness. Atoxyl and other arsenical compounds proved fairly effective here, but they were to cause neurological damage and blindness. Ehrlich then turned his attention to syphilis as a more fruitful field of research using arsenical compounds, since one of them, atoxyl, had already been found to be effective in its treatment. Well, by now, Ehrlich's research empire had greatly expanded, since his institute, now named the Royal Prussian Institute of Experimental Therapy, had been relocated to Frankfurt in 1899. In 1902, he established at his own expense a department devoted to cancer research. Then, in 1906, he was also, in addition to his work at the Institute of Experimental Therapy, appointed as head of the Georg Speyer House for Chemotherapy. This research institute had been founded by Franziska Speyer in memory of her husband, a Frankfurt businessman. It was built next door to the Royal Institute. In his dual role, Ehrlich now controlled every aspect of the activities of both institutes. His office was cluttered, piled high with books, journals, magazines, and papers that covered every space and left virtually nowhere to sit. Sometimes you can have quite a lot of sympathy for Ehrlich in this. Every day he wrote out detailed instructions for his staff on what he wanted them to do, with different coloured pencils on colour-coded note cards or blocks, which were then delivered to the recipients by his devoted porter, Kadewright. Only Ehrlich knew the key to the colour codes. He was an excitable and hard-working man who, according to his secretary and later biographer, Martha Marquardt, was driven by an inward urge, not from egotistic motives, but from a burning desire to help humanity. His life and work were like a candle burning at both ends. He smoked 25 cigars or even more a day. But his stamina and persistence was to stand him in good stead in his search for the magic bullet for syphilis. By 1907, he had synthesized more and tested more than 600 different arsenical preparations, assisted by the chemist Alfred Bertheim. The only member of the chemical department of the Georg Speyer House not to have walked out of the institute and protested Ehrlich's insistence that he knew more about the chemistry of arsenical compounds than the qualified chemists did. In carrying out these experiments, Ehrlich worked in his private laboratory at the institute. It was dominated by a central laboratory bench and a single Bunsen burner, and a clutter of bottles containing chemicals and test tubes. Now, that sort of clutter and chaos was not unique to Ehrlich's lab. But what was unusual was the absence of cylinders, retorts, funnels, beakers, bowls, or thermometers. Standard equipment in any chemical laboratory. Ehrlich preferred to use only the test tube when conducting his experiments. Ludwig Bender of the Casella Chemical Works styled him a virtuoso in the art of test tube experiments. Among the arsenical drugs tested was compound 606, which he set aside when his then research assistant reported that it had no effect whatever on infected animals. Although Ehrlich was cautious enough to have taken a patent out on it in 1907. Then, in 1909, he set a new research assistant to work on retesting the entire series of synthetic compounds for their action on the spirochetes, only to discover that compound 606 was actually very active against syphilis. You could say that Ehrlich was very lucky in his collaborator in this work. His former co-worker, Kitasato, had returned to Tokyo and was sending some of his best students to work at the most important European research institutes. In 1909, a bacteriologist was sent from Japan to work with Ehrlich. Sakahiro Hata, the 36-year-old bacteriologist, had already worked on inoculating rabbits with syphilis at the Kitasato Institute for Infectious Diseases in Tokyo, and was immediately set to work on infecting rabbits with syphilis and then injecting each arsenical compound into them. It was slow and meticulous work. But after a laborious series of tests on 605 compounds, he was able to report successful results with 606. Ehrlich wanted to make sure and insisted that the tests be repeated, just so he could be certain that they had found a magic bullet. But animal experiments were not enough. Human guinea pigs were needed, and 2 physicians volunteered to test the safety of this new drug. This was then followed by a series of clinical tests involving a group of doctors administering intramuscular injections of compound 606, by now named Salvarsan, into their most hopeless patients. This group was spread across the world, testing the effects of the compound in Bremen, Breslau, Magdeburg, Bonn, Sarajevo, St. Petersburg, Pavia, Catania, Zurich, London, and New York. Ehrlich remained the control freak and expected each doctor to follow his instructions exactly. And he was to give them very precise instructions on how Salvarsan was to be injected, and he warned them of the risk that the injection might induce fever if the needle was not absolutely sterile. Well, some of the results were amazing. Certainly, salvarsan was to be the first effective form of chemotherapy for the treatment of salvarsan. It replaced the formerly ubiquitous treatment with mercury. And by the end of 1910, over 10,000 sufferers from the pox or syphilis had been successfully treated with salvarsan. There was great demand for this treatment, and cures were seen as quite spectacular. In getting mass production underway, Ehrlich was fortunate in already having strong links with two local chemical companies, Casella and Meister Lucius in Browning at nearby Höchst. And new agreements were made between the Georg Speyer House and the two firms for the manufacture of the drug salvarsan. Ehrlich, though, insisted on retaining quality control. And one of the chemists at the Georg Speyer House, Alfred Bertheim, undertook much of the chemical work. But the main disadvantage of salvarsan was that it was still toxic. Well, what else did you expect from a treatment based on arsenic? Could cause abdominal pains, vomiting, convulsions, jaundice and skin complaints as its side effects. And in 1914, a modified form of salvarsan was developed from Ehrlich's compound 914, although Ehrlich himself was less enthusiastic about it and believed that the original compound 606 was superior. However, this newer preparation, neoarsphenamine or neosalvarsan, was much less toxic in its effects and more soluble, but because it was milder, required many more painful injections into the bloodstream before a complete cure could be effected. This was far from being the single shot of a magic bullet that Ehrlich had dreamt of. He had further worries about the progress of his drug when the First World War broke out in August 1914. Although he was a fervent German patriot, Ehrlich felt that no good would come from the war. Serum testing for the Imperial German Army now took priority over all other work at both the Royal Prussian Institute of Experimental Therapy and the Georg Speyer House. The chemist Alfred Bertheim, who had been central to Ehrlich's work, was called up by the cavalry. However, he caught his spurs on the stair carpet of the house in Berlin to which he was billeted, fell down the stairs and fatally fractured his skull before ever seeing active service. Contact with Ehrlich's scientific colleagues and friends in France and Britain was cut off. But he was able to continue with some of his work on silver salvarsan with the help of a new chemical assistant, Paul Ehrlich from Zurich, a young researcher who was to go on to win the 1938 Nobel Prize for Chemistry. Ehrlich was also concerned that in wartime conditions, his very precise and careful instructions on the administration of salvarsan might be ignored, and there would be many more adverse reactions to the drug than there already were. He was overworked, he was depressed, and he suffered a slight stroke in December 1914. However, he was to die from a more massive stroke in August 1915. Well, Ehrlich had certainly made his mark and was a very important figure in the development of chemotherapy, in the treatment of infectious disease with vaccines, in the understanding of how everything worked, and in the development of an effective treatment for syphilis. When a film was made of his life, it was called The Magic Bullet. It somehow skirts around the whole question of what the magic bullet was against. In the 1930s, there was a taboo on the mention of sexually transmitted diseases. But certainly, by all standards, Paul Ehrlich was a great scientist. He was affectionately remembered by his colleagues and staff. However, in the 1930s, Nazification meant that he was to be eliminated from the institutional memory of the two institutions he had headed in Frankfurt. However, luckily, many of the people who'd worked with him who were not Jewish hid away many of his items of equipment and anything they could find that related to his work. It was hidden away in a shed. Well, Ehrlich was Jewish, but probably would have considered himself German as much as anything. He was certainly patriotic. And fortunately, the fact that many of his papers were taken to North America by his family fleeing the Nazis, and the efforts made by workers in Frankfurt to preserve what otherwise would have been destroyed, meant that his memory lives on in Frankfurt. In the two institutes, or the successor institutes rather, to the ones he had headed. When I was researching the pox, the life and near death of a very social disease, I visited these two institutes and saw what they had, and also enjoyed spending time alone in a reconstruction of Ehrlich's lab. I've later taken part in various events organised to commemorate the various anniversaries connected with Ehrlich, organised by Fritz Sergel in Nuremberg. There were connections between Alexander Fleming, who was one of the first effective users of salvarsan in Britain, causing him to be nicknamed Private 606, because he was a private in the London Scottish Regiment, a volunteer regiment, territorial unit, and he treated many of his fellow other rankers with salvarsan. So a cartoon was produced showing him as Private 606, complete with A needle instead of a bayonet, syringe to administer salvarsan, and another syringe tucked into his stocking. Fleming had been given that sample of salvarsan by his head of department, Armjot Wright, Ehrlich's great friend and great sparring partner, who had been sent the sample from Ehrlich. He himself had no sympathy for any form of chemotherapy, and so handed it over to his juniors. So I do feel a lot of connection with the work of Ehrlich, and also have enjoyed the many times I've spoken about him, and also the opportunities I've had to celebrate his life. And we shouldn't forget that international cooperation was what it was partly all about, and indeed science still is. Well, I think we should certainly laud Ehrlich for his achievements, and also remember the principles of success in scientific research. Geduld, Geschick, Geld und Gluck. Patience, ability, money, and good luck. Ehrlich had all of those, and his achievements amply show that.